So hi, my name is Carlos and today I present the proteomics. So I will start to what is. The proteomic is the technology that investigates the proteome. And the proteome is the set of proteins that are present in a cell, tissue, and in any biological fluid in a specific time. Now, I will speak about the function of the proteins. So, the proteins have many functions. For example, in the first point or in the first function of the proteins is in the digestive enzyme. In this part, the proteins can help and facilitate the chemical reactions. The second function is in the immune function and in a specific in the antibodies support. The third function is support the regulation and the expression of DNA and RNA. Other function is support the muscle. Here support the contraction and the move of the muscle. The other function is provide support to the body, help the hormones to coordinate the bodify function, and finally the final function of the proteins is move important molecules to the body. So now I will go to speak about the proteins. So the proteins are macromolecules that are made up of amino acids. There is in total 20 amino acids that is amine, carboxyl, and variable chain in every amino acid. So they are part of all living organisms and are codified in the genomes. The proteins are important because they do the function of the organisms is the correct. And finally, the proteins are very important in the biomedical investigation because in a wrong functioning of it can make a disease. Now I going to speak about the medicine, proteins, and biomarkers. So most of the medicine that we have today are made for add or invit some proteins, and these proteins are biomarkers that are substances that can be used in the clinical setting to predictive, prognostic, or diagnostic disease. This should meet two requirements that are: first, that they be specific disease and be present, and second, be present in sufficient quantity to be detectable through an analysis. So, in fact, in many pathology search biomarkers that are proteins that are in our plasma and can identify disease in advance. For example, there is a investigation that is based on the proteomic. The principal objective is study the platelets and the blood plasma of patients with acute heart attack of myocardium to identify biomarkers that allows a better diagnostic that treatment of this cardiovascular disease. Now I will speak about the proteins and antibiotic resistance. So the bacteria in a future years have increased their resistance to the medicine and antibodies, and if this continues if the bacteria continues increase the resistance, it is estimated that by 2050 these same bacteria will be the main cause of death, causing about 10 million deaths per year. So today efforts are being made to make bacteria more sensitive to antibodies in order to understand with the proteome of bacteria how resistance is generated and make antibodies more effective. Now I speak about the separation, extraction and identification of proteins. So here we have a example of this topic that is this. So to know the proteome, the proteins must be extracted from the interior of the cell, separate to separate by their electric change and size, and they can be quantified and identified. Now I respond a question that is, why does the proteome constantly change? I see this question when I investigate this topic and I said that I like answer. And when I investigate this question have 5 answers that are the next. First one, the proteome change when we have a disease, infections, or change in the environment. In these situations, the body can produce more or less proteins and the proteome change. The second answer is in the production of cells. The proteins change for have the specific function and the proteome change. The third answer is the proteins are degraded by systems such as the proteasome and lysosome, and the proteome change with this degradation. The fourth answer is the proteins have processed and after the resistance. For example, phosphorylation, glycosylation, and acetylation, and the proteome change. And the fifth and final answer is when is in the old person. Here the body, the proteome change with the accumulation of the protein change. And finally, I like finish this presentation by a project that are created by the Human Proteome World Organization, that is the Human Proteome Project. This company, the factory, have the map of all the proteins of the organisms, and this is because the objective of this is can identify all the proteins produced of the human genome and all the variants, and with this can diagnostic of the disease will be easier. And with this I finish my presentation. Thank you for your attention, and we see in a other time.